carry information about the structure in the crystals. So I hope some of you made that connection, seeing the talk, the first talk in the morning. And the second talk was moving actually to larger scales. And the difference between this last talk and the one that I'm going to give now is in terms of the contrast. And probably you know that there are two major classes of contrast that one can use in imaging cells. And we call them intrinsic, meaning there is nothing added to the cell. It's just based on the optical properties of the cell itself. And extrinsic, which is mainly fluorescence-based. And you've heard a few minutes ago that actually GFP is kind of a way to combine the two. So you make the cell express actually something that is fluorescing. So in a way, the fluorescent signal becomes intrinsic to the cell. And that was a major breakthrough and Nobel Prize worthy. So my talk is about using no contrast agents and try to understand better how light interacts with a very transparent structures such as live cells. And it is my belief, I bet in my career on it, that actually there is a lot of room to do stuff without fluorescence. And I also believe that combining this kind of imaging with fluorescence is actually the key. So the method, my talk is quantitative phase imaging of intracellular transport. And I need to explain myself what all these words mean. So I will talk about quantitative phase imaging in general and in particular two methods that we use for imaging cells. And this talk is focused, we have different applications that we pursue in my lab, but I focus this talk on intracellular transport, and I'm going to show you all the results of membrane fluctuations just for consistency. Okay, so I'm in the Department of Electrical and Computer Engineering, probably you know that, and my lab is upstairs. You're going to see it today, I think. All right. First, let me make sure I have time to acknowledge the main players here. So Ru Wang is the one, the graduate student that's using a method developed by Joe Wang for looking at the intracellular transport. We collaborate with many people both on campus and outside, but for the cell transport, we work with the theorists from UCLA, Alex Levine, with a neuroscience group led by Martha Gillette. And Catherine, my wife, we keep collaborating on the red blood cells. That's been going on for a while now. So here's what we're trying to understand from a physical point of view. And I stole this movie from this paper. Incidentally, this is a GFP movie that is looking at actin. So to me, this is as a physicist, this is a fascinating movie, and what tells me is that how complicated things are. Where do you even start in trying to understand what's going on in the cell dynamically? And basically, my talk is about showing you the first baby steps that we think are in the right direction to try to scratch the surface of what's going on in here. So without knowing anything about cell transport, probably you can think about it and come up with the conclusion that there must be two types of transport. One, can you hear me without a microphone? How about that? Okay. So one is the random motion just due to the fact that we're at finite temperature. Okay? You know what Brownian motion is. You know it was discovered with these little particles of pollen in a suspension. Of course, the cell is at finite temperature, so we must have something like diffusion. And if you remember, I asked a question about the diffusion effect in the ion channel. And it turns out that diffusion plays a major role there. But at the same time, we expect that there is a second class of motions that are not due to thermal equilibrium, that are due to the fact, in a way, that the cell is alive. And because the cell is consuming energy, and is using energy, that means it's an out of equilibrium state. Okay? So synonyms for these words are, for random motion is equilibrium, thermally driven, Gaussian statistics, is this type of, the thermal equilibrium, the statistics is usually normally distributed, or passive motions. On the other hand, we have deterministic motions or out of equilibrium, ATP-driven, non-Gaussian statistics, usually, and we call that also active motion. So all these are synonyms in case you read about them or you hear about them. Let me talk about, that's a bad sign right here that my first movie doesn't work. There are some Brownian particles, so just picture those in your mind. This is the original paper by Brown from 1827. And what I wanted to point out here that first of all, he had a long title for his paper. He wasn't a very happy person. And also, he has the word active in his title. Okay, so blah, 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 on the general existence of active molecules. So, right from the beginning, I want to point out that what he calls active and what we call active today is different. There is nothing active about this, but think about the history of this discovery. So, he saw these particles in perpetual motion, and here's something that I thought was very interesting. Having found motion in the particles of the pollen of all the living plants which I had examined, I was led next to inquire whether this property continued after the death of the plant, and for what length of time it was retained. So the natural conclusion when you see something moving is that that thing is alive. So he wanted to check that, and he looked at dead plants, and of course he found that those are moving also. But nevertheless, he left the word active in the title. Now, okay, this is another movie that doesn't work. The theory for Brownian motion actually had to wait a little bit, and I don't know if you know this, but this is Einstein's most cited paper. Out of the 5 miraculous papers, the Brownian motion one is most cited. And this is a little formula that tells you if you track a Brownian motion in space, and you build what is called a mean square displacement, which means the displacement at t plus tau minus at t, you average that over t, you get something proportional to t, where the proportionality constant is the diffusion coefficient. Okay? So this is called the Stokes-Einstein equation, and it allows us to go from observing trajectory of particles to properties of the diffusion process, right here. So this will come in later. So the fact that this power law is unity when you plot mean square displacement versus time, that is a signature of the diffusion process. So in the back of our mind, let's remember we're trying to understand how much is diffusion, how much is active deterministic transport in a live cell. So that's what we do today. Particle tracking is actually a very well-established method. The idea is that you take these, in this particular case we have some beads, and they're flowing this way, they're drifting, so I can show you that they're not Brownian anymore. Therefore, the mean square displacement is curving up a little bit. So tracking the particles, it can actually tell you both about diffusion and active process. In this case, if you flow the particles like this, that means there's no Brownian motion anymore. Although locally it might have random fluctuations. So this curve of mean square displacement versus time will capture both. It will tell you at what length scale the motion can be assumed random, at what length scale is deterministic. So in this case, large scales is deterministic, local scales random. And here's an example where bias were from Urbana. There are many papers, high-impact papers on tracking, but this is a paper by Professor Paul Selvin's group, who's going to give you a couple of lectures, one tutorial on particle tracking and single molecule imaging, I think one of the afternoons, maybe Thursday. And the second on the science that those methods enable. So in this particular case, with tracking single molecules, they were able to find the actual way that the motor proteins walk by. And they found that they actually walk hand over hand, what it's called. And the steps is about 8 nanometers. So this is a very powerful, well-established method, tracking particles very accurately. What I'm trying to tell you today is that there is another method that we could use for the same purpose, completely different. And the idea is like this. First, the question is, how can we understand transport when the stuff that is moving is not necessarily discrete? Right? If you look at the cell in a phase contrast microscope, you see clearly some discrete particles, and then you see something that's not so discrete, that's continuous. That stuff is also moving. Imagine fibers of actin continuously remodeling in the cytoplasm. There is nothing to track. How do you deal with that? So that's one question that particle tracking cannot do. So our approach is this. We call it quantitative phase imaging, and the idea is to have, for instance, look at the plane wave. This is a plane wave incident on a cell. Let's assume the cell is homogeneous. There is refractive index n. There is refractive index n not outside. Because of that, as we learned already, there is a wavefront change. In other words, the part that goes through the cells is delayed a little bit. And basically, there is a lot of information that tells you the refractive index distribution of the cell in this wavefront. The problem with measuring this wavefront is that detectors are only sensitive to intensities. Right? So once you took the modulus square of the field, this phase information disappears. But we have ways now. This is quantitative phase imaging is actually an fast increasing field, in my opinion. It deals exactly with this, not only to measure intensity of the field, but to be able to measure the wavefront. Because that's important. All the information about the refractive index and the thickness of the cell is in this wavefront and not so much in the amplitude. The cells do not absorb, do not modulate the amplitude too much, but they disturb the wavefront significantly. That's the reason phase contrast microscopy works, another Nobel Prize idea. It's because Zernike managed to put the information in this wavefront back into the intensity. That's how the cell, from a very transparent, low contrast image, you end up with a high contrast image without adding contrast agents, just because Zernike coupled this phase information back into the intensity. So I'm going to tell you how we do this in a couple of ways. We develop many, many instruments to do this. It's quite challenging. But now I think we got it under control. But let's say we know how to do this. Then what? What do we do? Well, this phase shift is actually the product between the local thickness of the cell, h, and the refractive index contrast, n minus n naught. It has been shown more than 50 years ago that actually the integral of this, so if I were to, imagine this is a phase surface. If I integrate that, you get something that's linearly proportional to the dry mass of the cell. What's in the cell that's not water. Why? Because the phase information is actually the contrast between this and the surrounding medium, which is usually a solution just like water. So that's intuitively why integrating this phase information gives you the dry mass with very, very high accuracy. If you're able to measure this phase shift with 1 milliradian accuracy, you will be able to get the dry mass with a fraction of a femtogram. There is no instrument that can give you femtograms yet. I think the best you can do is picograms, many of them. So this has been known for a long time, that you just integrate this, you get something like dry mass. So that's another thing we're doing in the lab. We're looking very accurately at how cells grow. Look at this mass increase. But what I want to tell you today is about fluctuations. So what happens if I have a delta phi? If I have a fluctuations in the phase, that could be due to fluctuations in the thickness at constant refractive index. What is this? This means that locally the membrane went up and down, right? Okay, we're going to look at that. But there is also another contribution that could be at constant thickness. But now imagine there is refractive index fluctuation. How can that happen? Well, if a particle is passing by in the plane, in my image, I'm imaging from the top and this particle is passing by. At every particular point, I'm going to get the fluctuations when this particle is passing by, right? Like looking on a highway from above. So you see, this very optical quantity, and for most biologists, quite abstract, honestly, phase shift, now connects to something that people can understand and use. Thickness fluctuations, refractive index fluctuations, still abstract, but we're going to kind of decode. We see what that means. Basically, it means that any particle or any material that has different properties from the surrounding medium, we can call that a particle, and that will generate fluctuations in refractive index. So again, H, out of plane fluctuations. The cell sits here, out of plane fluctuations in the membrane. Refractive index, in plane fluctuations, in plane transport. Okay? Okay, so once we understood this, then how do we treat this? Here's where it gets interesting, and we're very happy to find a way to actually crack this down. Many of you work with optics and know about the dispersion relation. Basically, this governs the interaction of light with the material, but it can be applied in many other fields. In particular, in this case, the dispersion relationship tells you kind of the correlation time for each spatial frequency. So imagine now I have a membrane fluctuating, this left-hand side. It's fluctuating. It's like our ocean that I mentioned to you in the beginning, right? In the first lecture. You are on an airplane, you look down the ocean, you see these random fluctuations on the surface of the ocean. One way to describe that is decomposing into sinusoids, right? Decomposing it into Fourier transforms, spatial Fourier transforms. Now, what happens in general in a material is that each sinusoid, let's say of a particular period in microns, in this case, has a certain time scale. So intuitively, you can think of it this way. Larger fluctuations require longer time, right? As opposed to fluctuations that happen at short scales, right, are faster. The equation that connects these two, the temporal scales and the spatial scales, is called the dispersion relation. So going back to my membrane fluctuations, if I'm able to measure these membrane fluctuations, and I'm now plotting the inverse of that time, correlation time, which now becomes the frequency, versus Q, which is the spatial frequency, okay? So temporal frequencies versus spatial frequencies, I get these power laws. It just happens this way. It happens, as I mentioned to you, linear problems break down beautifully in the frequency domain. That's kind of the trick that nature plays on us. We're equipped with space and time detectors, but in fact the problem breaks down nicely in the frequency domain. So look at this. If I'm able to measure this omega versus Q relationship, and I get a linear relationship in Q, this is a sign that the membrane is dominated by tension. It's the same tension in a soap bubble or a droplet of water on a surface. If it looks like Q cubed, it's a sign that it's bending dominated. Okay, so each membrane has a bending modulus. I measure the fluctuations. I'm going to show you exactly how we measure those. I do this frequency, temporal frequency versus spatial frequency plot. I get Q cubed, and that coefficient that comes in front gives me the property of the membrane, bending modulus. There's another coefficient here that's less important. On the other hand, we do the same thing. So these were membrane fluctuations, but now we're looking at transport, particles going in the XY plane. If the temporal frequency is linear in Q, this is a sign that the particles move deterministically, like you taking a bus. Okay, that will be, if you write the dispersion relationship for you sitting on a bus of constant speed, it will be V times Q. Okay, where V is the velocity. If it looks like Q squared, that is a sign of diffusion. And right here, you kind of see what we're trying to do here. We're measuring something experimentally, and then we analyze it. And if it comes out to be Q squared, it means that whatever is moving is governed by diffusion. If it looks like Q, it's governed by something active. That somebody is giving energy to that system, is dragging it at a constant speed. There are other more weird terms that can occur. And I give you one here. If it looks like Q cubed. So you see the powers could be similar, but they mean totally different things, right? The physics is different. One is a membrane up and down. One is these particles moving around. Totally different. So Q cubed will be a sign of a chain. Imagine a polymer made of little monomers. This is diffusing. So in a way, the higher the power of Q, the slower the process. You see, this is fast. Directed motion, Q squared, slower. Diffusion is slower than directed motion, right? Q cubed, this is diffusion, but except of a single particle, now you have chain particles, and those, of course, are slower. Okay, so that's kind of to give you an intuition about this. Any questions so far? Yep. Yes.
And are you fitting them to the...
I'm going to show you exactly what we do. We are fitting them, yes. Yeah. Okay, the question is, what do we do exactly? Because I didn't tell you. I'm just giving you the principle. And she was right. What we do, we measure these fluctuations somehow, optically. I'm going to tell you that. And then we end up fitting some curves. We're going to calculate omega q. And we're going to look at them and fit them. And the coefficients of the fit will be something to do either with the membrane properties, mechanical properties, sigma, kappa, or properties of the trap. Yeah.
These processes are taking place in the cell at the same time. So there is fluctuation out of plane and in plane. So how are you...
Thank you for keeping me honest. I forgot to point out to this. That is absolutely right. You have both at the same time. And if you ask many people in the field, sometimes don't matter. Roughly, yes. So you don't need to assume anything. Okay, if there are no more questions, let's go eat. Thank you.